everyone, and welcome to today's webinar in the SERDP and ESTCP webinar series. My name is Rula Adib. I'm a senior principal at Geosyntax Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I will be facilitating today's call. The webinar will consist of a brief overview of SERDP and ESTCP by Dr. Andrea Leeson, followed by a list of upcoming webinars in the series. After Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features 3 presentations on the bioavailability of contaminants of concern in soils at DoD impacted sites. First, Dr. Hans Struve will present an overview of SERDP and ESTCP funded research on the bioavailability of contaminants in soils and key recommendations. Dr. Struve's presentation will be followed by a brief question and answer session. Then, Ms. Yvette Lowney of Alloy will present on polycyclic aromatic hydrocarbon interactions with soil and effects on bioaccessibility and bioavailability to humans. Her presentation will also be followed by a short Q&A session. Our third and last speaker, Dr. Nicholas Basta from the Ohio State University, will discuss mechanisms and the permanence of sequestered lead and arsenic in soils and their impact on human bioavailability. We will conclude the webinar with an interactive Q&A session with all 3 of our speakers. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand corner of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to please submit questions well in advance of the Q&A session. On this slide, we have provided a few suggestions in the event that you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line. However, if you continue to have problems, please submit a comment using the chat box in the lower left corner of your screen. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, and also the Program Manager for Environmental Restoration Program Area. Andrea has been with SERDP and ESTCP since 2001, and before that, she served as a research scientist at Battelle Memorial Institute, where she conducted R&D efforts on in situ bioremediation. Andrea?
Thank you, Rula. And before we jump into the webinar today, I'd like to give everyone a little bit of an overview about who we are. We're 2 programs. The first I'll mention is SERDP, which is our Strategic Environmental Research and Development Program. And we have been around for about 25 years as a partnership between DoD, DOE, and EPA. And we fund everything from fundamental to applied research with the goal always to impact the real-world environmental management. Under ESTCP, we have the Environmental Security Technology Certification Program, and this is our demonstration validation program in which we hope that the knowledge and the technologies developed under SERDP can mature and then be demonstrated under ESTCP. We have 5 program areas. 4 of them are under SERDP, 5 are under ESTCP. The Energy and Water program area is the only one under ESTCP. The rest, Environmental Restoration, Munitions Response, Resource Conservation and Climate Change, and Weapons System and Platforms exist under both programs and are managed by a program manager jointly. Under environmental restoration, we have several main focus areas: contaminated groundwater, contaminants on ranges, contaminated sediments, wastewater treatment, and risk assessment. And the webinar we'll see today falls under the environmental restoration program area. With the webinar series, we will be featuring projects from all of the program areas, and shown here are upcoming webinars that are planned so far. Our next one under the Environmental Restoration Program area will be on December 15th and is dealing with contaminated sediments. And after that, we're going to be going into a series of our award-winning projects from each of the program areas. Registration for some of these webinars is live, and we will announce these award-winning projects later on this year, probably by early December. You can always view information about our upcoming webinars at our link here. And at this point, I'll turn it back to Rula, and I hope you enjoyed the webinar today.
Thank you, Andrea. And it is now my pleasure to introduce our first speaker, Dr. Hans Rue. Hans is a technical advisor to SERDP and ESTCP. He has over 30 years of experience in the assessment and remediation of contaminated soil and groundwater. He was a principal at RETEC and HGL before starting his own consulting firm through Consulting LLC. He is co-editor of 4 SERDP facilitated books on in-situ remediation of contaminants in the subsurface. He received 2 bachelor's degrees in biology and soil science from Oregon State, a master's degree in soil science from West Virginia University, and a doctoral degree in soil science from Cornell University. And with that, turn it over to you, Hans.
Thank you, Rula. My job here is to give a brief introduction to this topic in general and an overview of some of the SERDP and ESTCP work in this area. Slide 16. In terms of what I'm going to talk about in my 10 minutes, I'm going to give a brief introduction to some of the terms and concepts that are important to understand the other talks in this area, give you a short overview of the status of SERDP and ESTCP projects on this topic, and then briefly cover some of the remaining research and development needs that have been identified recently from talking to experts in the field. All of this information and more is available in a white paper that has recently been posted on the SERDP website on bioavailability of contaminants in soils and sediments. Slide 17. A convenient starting place is the National Research Council's 2003 report on bioavailability of contaminants in soils and sediments. That laid the groundwork for using bioavailability and risk assessment, but it also recommended that there was a real need for a better scientific understanding and better tools to measure bioavailability. The figure here was a key figure in that NRC report. It illustrates the processes that occur in the environment that can reduce the amount of contaminant that's eventually absorbed by an organism. Note that strong sorption, the bound contaminants in the upper left that are bound to soil particles or sequestered within them, that binding, if it's strong enough, can have a strong effect on the amount that's eventually absorbed. Note also that process E there at the right are the internal processes within the organism that can also affect any deleterious effects. And finally, note that there are 4 different processes there that are identified, some of which occur outside the cell or outside a membrane, like the gut lining, that all can have their effects on the eventual amount of absorption. Next slide, 18. In terms of using bioavailability and risk assessment, the key parameter we have to worry about is so-called relative bioavailability, or RBA. And that's defined as the amount that's absorbed by an organism from the soil divided by the amount that's absorbed from the medium that was used in the original toxicity study. So for example, if you absorbed 100% in the original toxicity study when you fed mice or some animal feed or liquid that contained the contaminant of concern, and you absorbed only 50 when you gave the organisms contaminants on soil, then your RBA would be 0.5 and your eventual risk-based criteria would be twice as high. Next slide. So there is this direct relationship of the RBA, and it's important to realize also that when we use it in human health risk assessment, it can be used in either the exposure assessment or in the toxicity assessment. And the equations, I won't go through all the terms there, but the equations are show that the RBA is directly linked to the eventual exposure or toxicity. Next slide. Our first effort in this was to identify what inorganic constituents in particular would be most important for DoD to focus on. And Dr. Mike Ruby did this work and published it in 2005, where he surveyed a bunch of different databases and identified really the big five. Lead and arsenic are the dominant contaminants of concern in terms of metals. Chromium, cadmium, and antimony are also least present on more than 5% of the sites that exceed human health criteria. And in virtually all cases, exceeding those criteria was due to the oral ingestion of soil pathway. Next slide. The two terms that are really important to distinguish are bioavailability and bioaccessibility. So if in the figure in the left there, if chemicals are attached to soil and they're ingested by a human, they dissolve off of the soil particles into the gut fluid, and that's termed the bioaccessible fraction, the total amount that's accessible for eventual uptake. And we can estimate that by tests in a laboratory where we extract it in simulated gut fluid, for example. And that's relatively cheap. So a lot of SERDP's focus has been on validating in vitro tests that could provide a reliable predictor of the amount that will eventually be bioavailable. In terms of estimating how much is actually taken up by an organism, you have to do that in vivo, in live animals, and those are termed in vivo bioavailability assays. But in terms of cost, they're substantially more costly and can take a lot of time as well. So we're talking about a few hundred versus a few $1,000, and the time and cost involved in bioavailability testing directly are just make it impossible to use it in any routine way in doing risk assessments or in monitoring soils. Next slide. In terms of the status of some of the SERDP and ESTCP efforts, I've organized these by 5 of the key recommendations that came from a workshop that SERDP held in 2008. The first 2 are validating in vitro assays for arsenic and for DoD relevant organics. And Dr. Basta and Yvette Lowney are going to talk about the 2 projects highlighted in red there that relate to those workshop recommendations. In addition, SERDP in the past has funded some work on validating in vitro lead and arsenic assays and trying to predict bioaccessibility from soil properties. The third one there is that we were also tasked with developing some bioavailability guidance, and that work has been mostly done through ITRC. Those are the bioavailability of contaminants and sediments document put out in 2015. And SERDP is also cooperating with ITRC on a guidance document for bioavailability in soils, which is due next year, and the public page link is at the bottom there. Next slide. The 2 other recommendations I want to briefly touch on. There was one to develop valid soil limits for DoD contaminants that would include bioavailability. One of those was done by Dr. Roman Lano on ecological soil screening levels for a series of DoD-relevant contaminants. And Tom Trainer has done work on the mobility and bioavailability of lead and antimony on training ranges. And finally, there was a recommendation to develop remediation technologies based on reducing bioavailability directly. And the two projects there have involved looking at the use of phosphates to treat lead in particular, and lead and arsenic combined in some cases, and then iron-based amendments to treat arsenic in particular. Next slide. I'm just going to touch on the one to develop bioaccessibility predictions from soil properties. The work was done by Dr. Phil Jardine and colleagues, and looked, the two key graphs that I've included here show the effect of total organic carbon and iron oxide content on chrome and arsenic bioaccessibility, respectively. And the key point there to take away is that there is a strong effect, but it's difficult to develop robust predictions from the kind of data that you see there. But in combining a bunch of different properties, there have been at least screening level predictions that were developed and were used in a soil bioaccessibility tool that I believe Dr. Basta will touch on briefly. And next slide. In terms of the remaining research and development needs, there's a need to validate in vitro models for treated soils in particular. There's little data on soils that have actually been treated for lead and arsenic, and it's necessary to be able to measure the performance and also verify that it's going to be permanent in nature. There was a workshop recommendation to evaluate dermal exposure to carcinogenic PAHs because dermal exposure has become a much more important pathway potentially. And finally, there was a recommendation to validate the mouse model using mice instead of swine to test bioavailability in vivo. It's far less costly than using swine, but it still needs some validation. And finally, the last slide. All this information and more is available at the SERDP website under its featured initiative on bioavailability in both sediments and soils. And with that, I guess I can take questions.
Yes, thank you, Hans. We have a number of questions for you. You had one slide where you defined bioaccessibility versus bioavailability. Can you please elaborate about the similarities between the two terms and also the differences?
Sure. If we were able to truly measure and verify that we had extracted all of the bioaccessible fraction of the contamination, that would be a worst case value for how much might eventually be taken up. In most cases, I don't think we know very well what the relationship is between how much is bioaccessible, that is dissolved in the gut fluids, and how much is bioavailable, that is how much of that fraction is taken over the gut lining. And in practice, what we've ended up doing, I think, in most cases, is having in vitro tests that are designed to be predictive of the bioavailability. And so they may only measure a fraction, but by empirical correlation, they are valuable in terms of predicting how much will be taken up by a swine or mouse or human. So that the terminology is a little bit complicated and messy at times, but theoretically it's how much bioaccessible is how much can be dissolved into simulated gut fluid or real gut fluid, and bioavailable is how much of that fraction is taken over the gut lining and actually comes into the human.
Thank you, Dr. Stroh. Another question for you from one of our audience members. For inorganics, how important is speciation to bioavailability and bioaccessibility?
I'm going to defer to Dr. Basta for the most part, but obviously it's critically important, especially in many cases. And there are, both for lead and arsenic, there's a whole complex of mineral species that can impact bioavailability. but I think it's very hard to be able to say X species has Y bioavailability in all cases. It unfortunately has to be empirically determined, I think.
Thank you. And then you talked about or had a slide about the research needs for this area of work. How much is SERDP and ESTCP, how much are they doing to begin the process of addressing some of these needs, if any?
Well, that's a good segue to the next two talks, because both of them are going to touch on the first two in particular to some extent. At the present time, there's no separate effort to address any of those research needs, but as I said, to some extent they'll be touched on by Yvette and Nick.
Thank you so much, Hans. And we'll transition now to our second speaker. This next part of the webinar will be presented by Ms. Yvette Lowney, an independent environmental consultant focusing on human health risk assessment for clients in the government, industry, and also non-governmental organizations. Yvette has over 30 years of experience conducting research on the assessment and exposure and risks from chemicals in environmental media, workplaces, and consumer products. In the context of conducting risk assessments for contaminated sites, Yvette has designed and coordinated original research regarding the bioavailability of metals and organic chemicals in soils. Results from her work have been presented at professional meetings and also as testimony before scientific advisory panels and in peer-reviewed publications. In 2015, Yvette served as part of an international delegation to provide an advanced risk assessment workshop in the Philippines. And with that, I turn it over to you, Yvette.
Thanks for the introduction, Rula, and Hans, thanks very much for setting up some background information that's going to be useful as Nick and I go through some technical presentations here. So I'm going to talk about PAHs in soil and soil chemical interactions that affect human exposures. The presentation is going to discuss the impetus for the study, some of the key research components, our results, and the implications of those results for risk assessment. This slide, we're not going to go through all of the various components of this, but this slide presents a listing of some of the various sources of PAHs to soils. The point of this slide is that PAHs are not introduced to the environment as an isolated chemical. You know, it's not like we have a benzene spill. We never have a spill of benzoate onto soil. So PAHs are introduced to the environment in complex mixtures and with specific sources. And you'll see that that's really important as this goes forward. So our objective was to provide some information to update the EPA default assumption in risk assessment that assumes that 100% of PAHs in soils are bioavailable, or the RBA of PAHs in soils is 100%. As Hans explained, for PAHs, the critical toxicity study for benzopyrene was based on absorption from spiked food. So an RBA is going to be based on absorption from soil compared to absorption from spiked food. There is lots of literature that suggests that the RBA of PAHs is less than 100%. This little figure here provides an indication of the publications that have occurred over the years. There are nearly 24 studies done in vivo looking at the bioavailability of different PAHs, and double that if you include in vitro methods looking at bioavailability. But even given that broad literature, it is still limited because each of those studies is done on an isolated site. Some of them used unproven methods for estimating bioavailability, and many of the soil studies were contain high concentrations of PAHs that are not consistent with what we see in environmental situations. So our goal was to get some information that provides insights into the bioavailability of PAHs from soil, looking at the different sources, looking at the effect of different soil characteristics, and what the effect of concentration is. And our hope is with this new information and the tools that the research provides, we can decrease the uncertainty in the exposure assessments and risk assessments, and that can then affect the cleanup goals and risk estimates for those sites. So this is a schematic of some of the critical components of our research. We used constructed soil. I'll talk about that in a minute. And then we looked at soil chemical interactions and the factors that control the release of PAHs from soil. We did some physiologically based extraction testing at 2 different points in the research. We also looked at relative oral bioavailability and dermal absorption of PAHs from soil. I can't talk about all of these components, so this slide highlights the components that I'm largely going to talk about. So this slide is a depiction of the way we generated soils for our study. We chose to construct soils rather than use site soils. And we did that intentionally because we wanted to be able to control several factors. We wanted to be able to control the components of the soil. We wanted to be able to control the PAH source materials. And we wanted to know exactly what the concentration of the PAHs were that we put in. So this depicts the creation of the soils. We created an ASTM soil. Then we added sorbents, the things that the PAHs might adhere to or become bound with in the soil. And so we added coal dust to see the impact of that, and we reduced the peat with a natural organic matter, and we also changed the clay content. We changed the amount and the type of clay to see what the effects of that were. Then we mixed PAHs in either solvent, in a soot source, in skeet fragments, or in fuel oil. And we mixed that with the soils, weathered them through wet-dry cycles for 2 months, and then those served as our study substrates. So this table gives an indication of what we were looking at. So we started with a test soil, the ASTM baseline soil. Here we talk about what source materials we used for the PAHs. Across the top, it describes the different types of soil composition that we studied. And then these are the target concentrations of benzopyrene. The PAHs were spiked in a mixture, but the concentrations that I will be mentioning always reflect the target benzopyrene concentration in the soil. So concentrations spanning several orders of magnitude. The soil chemical interactions were assessed using aqueous equilibrium experiments. This method assesses the strength of binding between the PAHs and the soil components. We looked at a full suite of EPA priority pollutants. And one thing I want to point out over here is that I think people should look at so we will be recording KD, which is the equilibrium partition coefficient. This is calculated as the concentration that remains in soil divided by the concentration in water once the soil and water are at equilibrium. For those of you who are biologists, you might think, oh, a KD is a dissociation constant, and so a higher number means a higher dissociation. In this context, a higher number means a higher propensity to stay bound to the soil. And so that's important for interpreting the results I'm about to show. This slide contains a lot of information from our our assessment of soil-chemical interactions. This research has been published, so if you want the details, you can access it in our publication by Zia et al. A couple of things I want to point out here. So the horizontal bars and the pluses indicate the results from traditional equilibrium partitioning models. And I'm not going to go into detail on that. The short story is that the traditional models were unable to predict our observed results. We were able to improve on the modeled results with a multi-domain model, and those results are presented in the publication. The key point I want to make with this slide is represented in the diamonds. So the diamonds of different colors indicate soils. These all had the same 1 part per million target benzopyrene concentration, and with the PAHs introduced in different source materials. And as you can see, the KDs varied consistently across the different source materials for the different PAHs, with the KD value being the lowest when the PAHs were introduced in solvent. KD increased if it was in fuel oil, and then were quite a bit higher if the PAHs were introduced in either soot or skeet. The trend is very clear, and the magnitude of it is important when you look at the scale on the y-axis is a log scale. So this indicates that the soot and skeet as PAH sources resulted in a much stronger binding of PAHs to soil and a lower dissolution. One of the other key findings of this research involved the results that came out of our trials of understanding what happens if you add more black carbon to the soil. What happens to the bindings of the PAHs in soil? So on the y-axis in this slide, it isn't just the KD, but it's actually the KD in soils with charcoal added to the soil versus KD in soils without charcoal. In every instance, for each of the PAHs investigated and for each of the source materials investigated, the research showed that the addition of black carbon to the soils resulted in higher binding to soils for every pH source material. So the conclusions from this piece of the research is that PAH source is really important in governing PAH partitioning. We think that understanding the PAH source and contaminated soils can help risk assessors and people dealing with contaminated sites in assessing whether or not additional bioavailability investigation would be useful for the risk assessment process. The other major conclusion is that black carbon content can substantially increase the binding to soils. And this potentially has importance for the use of in situ remediation. We found that other soil characteristics had small or little effect on PAH partitioning. And as I mentioned, these results and details of the research can be found in Zia et al. 2016. So moving on to in vitro study, we've included in our research an animal study of bioavailability because in our regulatory environment, animal studies are required to demonstrate bioavailability and a difference in relative oral bioavailability from soils. We did this with the same range of soils with diverse characteristics and contaminant sources. And the other reason we wanted to have this is so that we could have a library of soils against which to assess an in vitro method for assessing bioavailability. And one of the main things in this effort was that we were focused on having an animal model that would give relevant data at environmentally relevant concentrations. In order to do that, we realized through several initial investigations that the best way to go at this was going to be to use a radio-labeled spike, benzopyrene. So as Hans explained, to get an RBA estimate, we needed to be able to compare the bioavailability of benzopyrene from soil versus benzopyrene in food. So we spiked food. We took our test weathered soils with the tritiated benzopyrene. We administered those to rats and collected the area under the curve of tritium in blood. And then we used the AUC from soil and divided it by the AUC from the spiked food to calculate bioavailability, or the RBA. This slide depicts the type of data that came out of the research. So an AUC plots the concentration of the contaminant in blood over time. This top line with the dots shows the results from administering food. That's the AUC for the food dose. And you can see when we administered soil, which is represented in the blue dots, we found a much lower AUC, much lower bioavailability. And when we administered the soil that had benzopyrene but also charcoal added to the soil, the bioavailability went down even further. So this clearly demonstrates the lower bioavailability of benzopyrene from soils when compared to benzopyrene in the diet. It also shows that the addition of charcoal further reduced the bioavailability. This is a summary table of our results. And the key things, so you're familiar with many of the components of this. On this slide, we have the source material across the top, then the soil characteristics along the side. The numbers that you want to concentrate on are the RBA values that are presented here. And these are all published by Roberts et al. Just a couple of months ago. The conclusions are that in soil, even a solvent spike, the RBA is about 50% compared to absorption from the diet. Looking at fuel oil, if fuel oil is used as the source of PAHs to the soil, the bioavailability increased with concentration. And at first that was interesting to note, and then we thought, oh, well, this is related to the fact that the concentration is increasing, absorption sites may be saturated, but more likely it's the fact that we are introducing the benzopyrene in the fuel oil source. So there is more fuel oil, and that fuel oil probably serves as a substrate for liberating some of the PAHs from the soil. Conversely, if PAHs are introduced to soil in soot, the bioavailability goes down with concentration. That seems counterintuitive until you realize that the increased concentration means that there is more soot in that system. So the 1 part per million benzopyrene from soot has 100th of the amount of soot as the 100 part per million benzopyrene soil. And so with the higher concentration, you also have higher soot in the soil, which serves as a substrate for binding of the benzopyrene to the soil. The other thing that is illustrated here that is consistent with our earlier research on the soil chemical interactions is that when you add charcoal to the soil, the bioavailability is significantly decreased relative to the lack of having charcoal in soil by about a factor of 3 or greater. So the conclusions of the in vivo study are that we found an animal model that provided reliable data. I would say we generated an animal model that provided reliable data from environmentally relevant concentrations. The results indicate that the PAH source material has an impact on bioavailability as measured in this animal model. And consistent with our earlier work, the addition of charcoal resulted in reduced bioavailability. Finally, I want to touch very quickly on efforts to use these soils that have been fed to animals to see whether there are in vitro methods that can be used to predict bioavailability. So we took the 15 soils from the RBA study and investigated them in 5 extraction conditions. Those are depicted here. I'm not going to talk at all about these last 2. The short story with those is that they were not predictive, and so I'm not going to spend time on those. But I did want to present information that showed the results of the modeling of whether physiologically based extraction testing was predicted. So in this is called an in vitro to in vivo correlation. In vitro data are plotted on the x-axis, and the bioavailability for the matched soil is plotted on the y-axis. And what that does is if we have in vitro data, we can then use this relationship to predict the bioavailability. So the indication from this physiologically based extraction test is that it gives pretty good predictability. There was a soil that is clearly an outlier that needs to be investigated further, but it shows very promising effects for using in vitro methods. We also used a simple solvent extraction based on some literature information that butanol may be a good solvent for predicting bioavailability. And consistent with the literature, we found that that is a good predictor. It is not physiologically based, but to the extent that it is correlated with the animal research, we think that that provides a really good method for estimating RBA. The final one that I wanted to discuss is the 3550C. That's EPA method 3550C in comparison of that to bioavailability. This is an EPA method that is frequently used for assessing the concentration of PAHs in soil at contaminated sites. And what we found is that most of the time, so for 10 out of 15 of our soils, it overestimates bioavailability. But for 5 of the soils, I'm sorry, no, it says 6 here, that's a typo. For 5 of the soils, it actually underestimated bioavailability. And that's going to be important thinking through application of bioavailability adjustments in the future. So the conclusions are that our library of soils provided us with a strong basis for looking at an in vitro to in vivo correlation. The results indicate that both physiologically based and simple solvent extractions may provide useful indicators of bioavailability. And the overall research of the conclusion of the work is that across the soil chemical interactions component and the animal research, it's very clear that the sources of PAHs to soils are controlling bioavailability of PAHs from them. The black carbon is important to controlling the bioavailability, and that has potential implications for soil amendment technologies. And we think that in vitro methods show good correlation with bioavailability. There currently is no regulatory standing for using these particular in vitro methods, but at this point we think they're a useful screening method for sites and could be very informative to site managers. I need to acknowledge that this work was done by a large group of researchers across several organizations, and we appreciate all of their efforts. And with that, I want to encourage you to get more information. Let me know if you'd like any information from me. And we have submitted a final report on this project, which should be up on the SERDP website soon.
Thank you, Yvette. We have received a lot of questions for you. So we're going to start off with a question from the British Geological Survey. Can spiked soils ever be a good analog for soils from a contaminated site?
That is a very broad question. Ever is I would say yes, they can at some point. Our goal here was not to simulate a contaminated site. It was to provide basic information about what is it in soil that is controlling bioavailability, and then also to provide the library of soils that can be to support an in vitro method. And the goal was that then we would have an in vitro method that could be applied to site soils. The reason we did that is that in order to get reproducible, reliable animal results, we found that we couldn't do that with not radiolabeled soil and get meaningful results at PAH concentrations of environmental relevance. And that's been shown in additional research that has just come out recently, that to get the reliable animal data, the concentrations of PAHs in soils have to be quite high.
Thank you, Yvette. A question from the Connecticut Department of Energy and Environmental Protection. Can you please elaborate on what you mean by PAHs mixed with solvents at contaminated sites? I think that refers back to your sources slide.
Right. So basically, as a positive control, we use the baseline synthetic soil, and we spiked it with a mix of the 16 priority pollutant PAHs in solvent, and then just spike that into the soil. So that was a form of a positive control.
Thank you so much. Question from EPA. Do you have any future plans to build any fugacity models with this material?
So the fugacity models would go to the soil chemical interactions component of this work. It's this different kind of language for the same concept. We think that the results from the animal data here are very consistent with the results that were found in the soil chemical interaction component of the work. And both of those pieces need to be, a future piece is to work to harmonize the two components of that research further for better integration of the two pieces. I will say that the Zia et al. Provides a multi-compartment model, a multi-domain model that improves the partitioning results, and I think that might be what you're asking for here.
Thank you. Another question from the U.S. Army Edgewood Chemical Biological Center. What soils were used in the in vivo study? Were any natural field soils used? And if not, was this planned?
Rilla, can you repeat the very end of that question? If not?
Yes. If not, was it planned not to use any natural field soils?
Yeah. So I think I've addressed that and with some of the previous questions. We designed this work to be able to provide meaningful estimates of bioavailability at very low concentrations. And we couldn't do that with contaminated site soils because we would have had to use much higher concentrations. We also were not trying to characterize the bioavailability from a particular site because we decided we could, if we had picked a site, then we would generate data for that site. And if it didn't agree with another site, we wouldn't have a basis for understanding why. This was more basic research intended to understand what are the factors in soil that would be controlling bioavailability across different sites. But we did not use site soil.
Thank you. We have time for maybe one long question or two quick ones. So let's start with this one. How do you know that the PAHs were associated with soil particles versus the soot or skeet particles used to introduce the PAHs? Were the soot and skeet particles somehow separated from the soil particles,
or were they all analyzed together?
Yeah, that's a good question. So in the animal, in both major components of this, we were less interested in understanding whether the PAHs remained bound to the source material or whether they partitioned into other parts of the soil. Then we were interested, sorry, that's not constructed very well. We were more interested in understanding whether they dissolved and whether there was dissolution from the solid material into solution or from the solid material into animal tissues. We didn't care as much about where it was in the soil.
Thank you, Yvette. And one last quick one. Why is it that you included skeet as a PAH source in the soil chemistry part of the research, but you did not include it as a source in the animal study of PAH bioavailability?
Good question. So the soil chemistry component of this research used unlabeled materials. And so because skeet is very important in DoD contaminated sites, the work was funded by DoD, we really wanted to include an assessment to the extent we could of the nature of binding of PAHs in skeet contaminated soil. However, because we had to use a radio label in order to achieve the other goals in the animal research, we didn't have a skeet source that was radio labeled. It turns out skeet is very easy to manufacture. We found somebody who would make radio labeled skeet for us, but the cost of generating that was prohibitive. It would have nearly doubled the research budget for the animal piece of the work. So maybe that's something we will look at in the future as this moves forward.
Thank you, Yvette. On slide 34, this is a question from Alberta Environment and Parks. On slide 34, you indicated that coal dust was added as a geosorbent. Why did you use this material, and could it possibly be a source of PAHs in itself?
So charcoal is known to sorb organic contaminants. And so it is a very strong sorbent. And so that was, the goal was, it also has been used in bioremediation of sediments, and so and been shown to help prevent, help decrease bioavailability of contaminants in sediments. So we thought it would be a good substance to test in our assessment of sorbents. And we did have to look for a clean source of charcoal, and I can give you information about that if you contact me directly. The source material will also be described in the Gia et al. Publication.
Thank you so much, Yvette, for a fascinating presentation. The final part of today's webinar will be presented by Dr. Nick Basta, a professor of soil and environmental chemistry and also the co-director of the Environmental Science Graduate Program at The Ohio State University in Columbus, Ohio. Nick's research focuses on environmental chemistry and bioavailability of contaminants and nutrients in soil, with emphases on human, agronomic, and ecosystem health. His research program is internationally recognized. He has more than 400 career publications and has served on several editorial boards, including 12 years as technical or associate director for the Journal of Environmental Quality. Dr. Basta is also a fellow of the Soil Science Society of America and the American Society of America. Nick, I turn it over to you, please.
Okay, thank you, Rula. Can you hear me now?
Please.
Okay. Good morning, afternoon, evening, wherever you may be. I'm very glad today to present the results from our SERDP study ER 1742, which is Mechanisms and Permanence of Sequestered Lead and Arsenic in Soil. Today I will be going through this presentation outline, basically why use bioavailability, mechanisms of sequestered arsenic in soils and impact on bioavailability, long-term permanence of lead remediation, conclusions from the study, and some comments on validation of in vitro methods for DoD sites. So before I start, why use this? Why use bioavailability and assessment? And it's to prevent what you see in the picture in front of you, to prevent very costly, unnecessary remediation action, and I would say ecologically destructive excavation and replacement of soils. Many times, this is the most expensive remediation you could perform. Excavation and replacement is one of the most costly drivers of remediation. It can range from $200 to $500 a ton, and that's a lot of money when you think there's 1,000 tons in an acre. The second reason is remediation cost savings can be huge. Dr. Mark Richardson of Health Canada delivered a presentation entitled Bioaccessibility and Bioavailability, the Great Opportunity Not Realized So Far, at a special symposium on bioavailability we organized at CTAC in 2014. And he showed this slide. Basically, he calculated that the average remedial cost savings needed in Canada is $564,000 a site. The cost overrun, meaning the savings needed for all the sites in Canada, would be $2 billion. But if people would apply bioaccessibility, bioavailability testing, they could save $2 billion, essentially covering all of the cost overrun for remediation of all the sites in Canada. I'm not sure what it is in the U.S., but it's probably a bigger number, and the savings would be a bigger number. The specific objectives of the objectives of our research were targeted at barriers for use of site-specific bioavailability. These key questions were targeted. Which laboratory tests can be used to predict bioavailability for DoD soils? And secondly, are in situ treatments of lead-contaminated DoD soils, including fiery ranges, effective over the long term? Our specific objectives was to evaluate 5 cost-effective laboratory methods to predict site-specific bioavailability for adjusting exposure and risk assessment, and to evaluate the long-term permanence of lead-contaminated soil remediated by phosphorus amendments. The first part of our research project focused on arsenic bioavailability in soil. We evaluated 27 soils from a wide range of contaminant sources, locations, and sites, mining, pesticide, etc. The form of arsenic in the soil was determined by arsenic speciation. We measured arsenic bioavailability from juvenile swine and mouse dosing trials. We used 5 laboratory methods to measure arsenic bioaccessibility, and then we looked at results from all these studies were cross-correlated using statistical analyses. The second part of Project ER 1742 focused on the long-term permanence of lead remediation. We evaluated key soil, chemical, and microbiological processes that could undo in situ remediation already performed at these 2 sites. One site was an EPA site, which had a lead smelter contamination problem, and the other one was a DoD site, which was from lead firing range site. So the first part of the study on arsenic. We actually had 27 contaminated soils from a wide ranges of sources. We really worked to get a range of sources. And you see the sources from pesticide, orchard, mining, glass making, smelter, etc. The concentration had a wide range from 162 to 12,000 part per million. I'm going to show you some of the results now, specifically the arsenic speciation results. RBA is on the y-axis, and the type, and you can see this PCA is showing us the type of species we found. The take-home from this slide is arsenic speciation and soil type greatly influence the bioavailability of arsenic. Bioavailability range from high calcium arsenate, iron arsenates, to very low bioavailability for arsenical sulfides. Arsenic was strongly absorbed to soil and had medium to low bioavailability absorbed to iron oxides on soil. These results were consistent with other research that had been done previously in Canada. We used 5 in vitro methods previously correlated with bioavailable arsenic. These were the US EPA 9200 method, which is the same as the SBRC method in Australia. I will just call this the glycine method rather than repeat that. OSU IVG method, the UBM, which is the Unified Bioaccessibility method that was developed by the Bioavailability Research Group of Europe and is used in the EU. And this method, I've heard just recently, has ISO designation. The RMC, Royal Military Canada PBET method, and the California Bioaccessibility method. The amounts that we extracted, the amount of arsenic we extracted with these methods really varied. CAB was the largest concentration extracted, and glycine was the lowest. So these are showing the results of the in vivo, in vitro correlation, the linear regression, determining whether or not these methods could predict RBA or relative bioavailable arsenic. The top table shows the linear regression for the 5 methods using the mouse model to determine the RBA. The bottom table shows a linear regression for the 5 methods using the juvenile swine method. If you look at the table, you would see that both for mouse and swine, all the methods predicted bioavailable arsenic for most soils. They were all pretty good. The R-scores are a little higher for the mouse. There's a little more variability associated with the in vivo data with the swine. But take home from this is all methods predicted bioavailable arsenic for most soils. And I want to move to the second part of the study, which is the long-term permanence of lead remediation. In situ soil remediation using phosphorus soil amendments has been used successfully on many lead-contaminated sites, including DoD firing ranges. This type of approach is a bioavailability-based remediation where phosphorus soil amendments react with soil lead to form very insoluble lead minerals that have very low bioavailability. If these particles are then ingested, you would be reducing lead exposure and risk because they have lead bioavailability. So this has been demonstrated. What I'm showing here is a picture here at Camp Stanley in a previous ESTCP project showing that treatment of the site with firing range with fish bone, which is the phosphorus reduced bioavailable lead. One of the regulatory barriers are, how permanent is this remediation? How long will this last? Will the stabilized lead be released over time? That's what we looked at in our project. So in our project, we basically looked at remediation methods, and our conclusion was that remediated lead was stable. We looked at microbial fungal treatments. They had no effect on bioavailable or bioaccessible lead. We looked at chemical processes, including soil pH, acidity. We acidified these soils to very low pH. No effect on the stabilized lead. So we had similar results for both the Oak Ridge firing range and the Joplin lead smelter lead remediated soil. The lead in these sites were stable. And the Joplin site is probably about 15 years old now, going on 20 years old. So it's a long-term remediation. We at first started using, we asked this question too, can we use in vitro methods to predict the lead in these treated soils? Can we look at the reduction in bioavailable lead due to soil amendment, for example, using the standard method, EPA method 1340? US EPA method 1340 has been shown to be an accurate predictor of lead bioavailability in untreated soils. Can it be used in treated soils? Our findings show that 1340 was not an accurate predictor for phosphorus-treated soils. Phosphorus treatment reduced bioavailable lead determined from the mouse studies. So if you actually look at this figure, you can see method 1340 at pH 1.5, that's the standard method. Look at the black bars. And if you look at P treatment, you'll see that the black bar is around 30%. And then if you look at the mouse RBA, which is the last crosshatch, it's much lower. So the mouse RBA, the mouse bioavailability, was very low when the mice were dosed with this soil. But this is the predicted RBA from method 1340 at pH 1.5, and it's greatly overestimating the RBA. However, if you change the pH from 1.5 to 2.5 for the standard EPA method, it's almost the same as the mouse RBA. So if you extract the pH, US EPA 1340 accurately predicted the RBA lead in the treated soil. So we believe that using a modified method 1340 with a pH 2.5 should be investigated for P-treated soils in the future. So the conclusions for the study: IVABA bioaccessibility methods provide reliable estimates of RBA arsenic for use by DoD managers. Lead in phosphorus-treated soil is stable, and soil amendment with P is a long-term remediation practice for lead soils on DoD sites. But current measured in vitro methods, for example, 1340, may not be accurate to evaluate the success of these P treatments and performance using phosphorus remediation. And they may have to be modified, maybe increasing the pH or something, to become an accurate predictor of the animal RBA. So acceptance and guidance for using in vitro bioaccessibility methods to predict lead and arsenic for site-specific risk assessment is really growing. This year and next year are going to be banner years in this area. Just recently, the state of California recommended methodology guidance for site-specific bioavailability in California. You can now use the California bioaccessibility method to predict arsenic bioavailability for use in risk assessment in California. US EPA, the in vitro method 1340 that was approved for lead in 2007 will likely be approved for arsenic soon, and it will be termed EPA 9200. There's a major ITRC effort underway. We are in the middle. We just created a draft document on bioavailability and contaminated soil to assist U.S. state governments to implement bioavailability. This is going to be a really landmark document. We're going to be giving an overview of the whole topic, plus PAH, arsenic, lead, and other case studies that would be helpful for risk assessors. That's expected November 2017. So some risk assessors on the line here may be wondering, how accurate are these inexpensive in vitro methods of predicting lead or arsenic? Can I use it on a new site? Can I use it on my site? Well, to answer that question, you may want to do some further validation. And here's a mechanism. This slide shows a 4-step validation study to answer the question for a new study site soil. that wasn't in the data I just showed you. You could collect your soil, measure in vitro at your new soil, in vitro bioaccessible metal or metalloid. You then take that in vitro number and plug it into the published IV-IVC regression equation that I just showed you for the whatever method you're using, and that will give you a prediction of RBA arsenic. So now you have a predicted RBA. Then what? Then you measure the RBA arsenic. I have listed here juvenile swine dosing, but you could probably use mouse dosing. If that's really going to be up to the regulatory body you're dealing with. So you would actually do the animal study, get your predicted number from in vitro, and see if they compare. And if they compare, you're good. This ESTCP project ER 200-517 already did this. They actually this project was entitled "The Effect of Soil Properties on Toxic Metal Bioavailability, Field Scale Validation to Support Regulatory Acceptance." The PI was Dr. Amy Hawkins from NAVFAC. We did all the soil bioaccessibility testing, and Dr. Castile did RBA determinations using his juvenile swine model. So I want to show you some results. There were 2 sites that we did validation studies on, 2 soils. So the top table shows results from a DoD site that was contaminated with lead, and the bottom table shows a DoD site contaminated with arsenic. So we collected the soil, measured in vitro, got the regression equation, plugged it in, compared it, fed the soil to the juvenile swine, compared the results. The juvenile swine RBA for lead in the top table, 99%, 90% CI, 70 to 127. The RBA that was predicted using the in vitro result, the cheap and expensive test by EPA method 1340, was 70.3. It's in the range. It's predictive, 70 to 127. We used ROSU IVG. We hit 102. That was right on the money. So that was good news. It shows that the in vitro methods accurately predicted the juvenile swine RBA lead. At the other site, for arsenic, the bottom table, we actually dosed the swine and got 14% relative bioavailable arsenic. Range was pretty tight, 13 to 15. And both the OSU method and the US EPA 9200 method predicted the RBA arsenic for the swine. So soil amendments can be used to reduce bioavailability to an acceptable level at contaminated DoD sites. In vitro methods are a fast and inexpensive method to predict bioavailability. Can we use in vitro methods to predict the success in treated soils, the reduction in contaminant bioavailability in treated soils? We're not sure. I think this is the new frontier. Research is needed to evaluate these methods and maybe see if these methods will work for treated soils, such as lead treated with phosphorus-containing soil amendments. This is very important because I believe that many times you're only going to be able to achieve a great enough reduction in bioavailability by treatment of the soil. Just some soils will have to be treated. And I included this picture because it is the 50th anniversary of Trekkies, Star Trek. This is for all the Trekkies out there. Hard to believe it's 50 years. Acknowledgments. I'd like to acknowledge all my project co-PIs. A lot of people worked on this project. Dr. Karen Bradham and Dr. Dave Thomas at RTP provided bioavailability data for mice. Dr. Cheney, Dr. Mike Fela, Dr. Phil Jardine, Dr. Chris Schatt did all the fungal work. Dr. Shekel did all the speciation work. He's not here, but Dr. Anderson did a lot of the statistical work, and Dr. Castile did all the juvenile swine work. And we did all the in vitro work, for most of it, not all of it. Okay, so with that, please visit the site for further information.
Great, thank you, Nick. We've got a number of questions. First one from the University of Florida. Among the 5 methods you tested against arsenic RBA, which in vitro method do you recommend based on your data?
I think they're all predictive. For most soils, they're all predictive, and so our study basically says that, you know, we recommend all of them for the study soils. There are some soils that some methods work better than others, but that gets down to specific soil types. The California method tend to work better on high iron soils that we found in California. It was more predictive, where some of the other methods were underestimating RBA arsenic. But in general, the materials that we ran into, all the methods were predictive.
Thank you. Another question from the Massachusetts Department of Environmental Protection relates to translating your observations to reliably predicting bioavailability. So the author of this question points out that one difficulty in doing this is that organic carbon and iron oxides are typically present as coatings on soil versus being uniformly present in the soil matrix. So the question is, if the bulk of organic carbon and iron oxides are concentrated on only 10% of the soil, is it possible to underestimate bioaccessibility by a factor of 10?
I don't think I quite understand that question. I mean, I realize they exist as coatings on soil surfaces, but these are real soils we used, and they weren't spike soils, so they probably had the coatings already in the soil. So I'm not quite sure how we were. I don't think we could be underestimating anything. We actually measured the bioavailability of the arsenic that was probably attached to coatings.
Thank you. A question from EPA. What is the primary reason to make you believe that pH adjustment makes 1340 more accurate?
Well, the in vitro method should be able to predict an RBA that reflects what's happening in the animal. So the lead-treated soil showed a large reduction of RBA in the animal. The pH 1.5 US 1340 method did not show that reduction. So it's not predicting what the in vitro method is only as good as it's only good if it can predict what we see in the animal. We dose the animals with the treated soil, big reduction. We dose them with pH 1.5. We do the prediction with the EPA method at 1.5, and we get virtually no reduction. So that's telling me that it's not an accurate predictor. However, we adjust the pH to 2.5, and we predict accurately what the animal is seeing. It's that simple. I should mention, if you go back to the 2007 document where the 1340 was developed, there was actually, that method was evaluated at pH 1.5 and 2.5, and both were correlated with RBA lead for untreated soils. So in fact, if pH 2.5 was used, selected as the standard method, it would work for untreated and it would work for treated, and that's much better than having a method that only works for untreated.
Thank you, Nick. A question from the U.S. Fish and Wildlife Service. Does the source of lead matter? For example, shotgun and small arm ranges versus lead from smelter emission.
Yes, the source absolutely does matter. And so there's two things. There's the chemical speciation, the form of the lead, so you're dealing with their metallic lead versus something that might be weathered lead carbonate at a firing range, and the lead carbonate would dissolve very easily in the acid in the stomach, where the lead shot wouldn't dissolve as quick. And then the second thing are the kinetics. We tried to mimic the shot or the ingested lead only stays in the stomach for 1 to 2 hours. So you you're trying to mimic what happens in the animal, and the shot, as you know, probably wouldn't dissolve as readily as the weathered firing range stuff or stuff at the smelter that might be lead carbonate. So, I mean, we actually have lead speciation data. I'll tell you, the lead speciation data was related to the RBA in the animal. It was pretty good. It explained 46% of the variance. It was bordering on almost being predictive, but honestly, the in vitro methods do a much better job. We get 70, 80, 90% of the variance explained with the in vitro, but still, speciation shows that the form of the lead really matters.
Thank you, Nick. I have a question that I'd like you to answer first, and then perhaps ask Yvette and Hans to weigh in. Is in vivo bioavailability testing commercially available? And if so, where can you get it done, and what would the cost be?
I can tell you, I can't give you a comprehensive list, but I can tell you I can count all the places on my one hand or two hands. And I don't need my toes. There's not that many places that actually conduct the animal studies. They tend to be expensive because they're very involved. The juvenile swine studies could run from $20,000 to $40,000 of soil. And that's because there's a lot, the design is very complicated. You have 36 animals, you need qualified people. It takes 1 month to conduct the study. So it's an expensive test. It's very accurate, but it's expensive. The mouse, I have no idea what the mouse runs. I've heard it's less expensive. I'm sure it is. But I don't know any commercial labs. But that doesn't mean they're not out there that are doing the mouse. In vitro methods are under $200 a test. So that's why the in vitro methods, I think, in the future are going to be used, because they're easy to run, there'll be more labs offering them, and they'll be inexpensive, versus the animal studies, which are much more expensive, and it's hard to get access to get. Even if you have the money, you may have to wait in line to get that test done.
Thank you. Yvette, would you like to weigh in?
Yeah, I think Nick hit most of the important points. The reason that we are trying to develop in vitro methods is partly because of the cost savings. There are issues with animal research as well. A couple of years ago, I did a study where I investigated the cost of doing arsenic bioavailability in different animal models, and at that time, it would cost about $70,000 to put 3 soils through either swine or monkeys, which were the 2 models at the time. So it's quite pricey. Since then, this rodent model has been developed. The hope with that is that it is more cost effective. That's a model that EPA has developed for arsenic. So they are available. They tend not to be with commercial labs, although I've heard of one recent PAH bioavailability study where the research was done at a commercial lab. And I could get you information about that. I'm not exactly sure which lab it was. So commercial labs are available. Mostly this work is being done in academic and government labs.
Thank you, Yvette. Hans, anything to add?
No, that's not really, although I have to say that if you want to use this kind of testing to make a difference in characterization or monitoring a site, only in vitro testing will let you do that. You can only test, you have to test a lot of samples to make decisions about what to do with things. Only that the question is, how much uncertainty are you willing to live with in the in vitro test that is reasonably predictive of the RBA? That I think is the question people really have to wrestle with. The methods should be available and transparent, and any analytical lab should be able to pick this up for in vitro extractions, frankly. Can
I add something?
Please do.
we just completed a study as a subcontractor for a DoD site, and what they did was pretty innovative. They said, well, what we want to do, it's a big site, so we'll collect one sample, make a composite, and get an animal number, an RBA, right? So pay for one animal. But we want to get an idea of the variance in the RBA to bioavailability across the site. So there we'll use in vitro methods because they're inexpensive. So you can use these in combination. The RBA will, you can have a real RBA through the animal model, and then you can use the in vitro method and get predicted RBA to get an idea of the variability across the site. That was the first time I saw that design that way. I thought it was pretty innovative.
Yeah, this is Yvette. That is a good study design, and we've used that for lead-contaminated sites, lead and arsenic-contaminated sites. And another way to do it is to nest them, not just decide you're going to do them all at once, but you have your site characterization data, then you can do the inexpensive in vitro assessments and understand your variability across the site, but also the likely magnitude of a bioavailability adjustment and whether or not it's worth pursuing an animal study. And so if your in vitro data indicate good reason to expect a lower bioavailability, you can then use that to hone in on which soils to select for animal testing or which to composite for animal testing, and also whether you have different sources that you need to characterize in the animal test. So definitely there is a way to use in vitro and in vitro data to inform your process for in vivo studies.
Thank you, Yvette. And then one last question before we conclude for the three of you. Starting with you, Yvette. Have you been involved in gaining regulatory acceptance of RBA in HHRA? And if so, what are the critical factors that affect success?
So I've been involved in several projects where we use site-specific data to get bioavailability adjustments for contaminated sites. I think that the two primary things that are important in having that happen are really good communication and a very good relationship with the regulators. And those two things obviously are related. So the regulators frequently need to understand the bioavailability sciences, what's behind it, why they should incorporate bioavailability adjustments, and to understand that it's not just a sneaky way to get a higher cleanup level, that it actually is about generating more information for your site and using that information to decrease the uncertainty in the exposure assessment and in the risk assessment, and therefore providing a more site-specific and more appropriate value for this site. So I've walked into projects where the regulatory community had never considered bioavailability, and lots of education was involved. Certainly going back and forth is involved. And then with in vitro methods, I think that they are best used as part of a weight of evidence evaluation. So, for example, although there is no approved arsenic except in the state of California and Hawaii, there's no regulatorily approved in vitro method. But that doesn't mean that you can't use in vitro data to get an RBA adjustment in your risk assessment, as long as it's part of the weight of evidence evaluation and lots of communication.
Thank you, Yvette. Nick, do you have something to add?
No. I will mention that the UBM method that has just gotten ISO designation. I know Mark Cave is on the line from the They developed that in the Bioavailability Research Group of Europe. That's been predictive. It's a good model for lead, arsenic, cadmium, and antimony. It's been shown to go beyond the metals that we've generally studied here in the U.S.
Thank you. All right, well, we're out of time, and before we wrap up, I would like to remind everyone that our next webinar is in 2 weeks on Thursday, November 17th. It will cover novel coating systems for use as high-performance chemical-resistant powder top coats. The webinar will feature 3 speakers, Mr. Mark Waitias and Dr. Beth Ann Pearson from the Sherwin-Williams Company, and Dr. James Wynn from the Naval Research Laboratory. Registration is open for this webinar, so please visit the SERDP and ESTCP webinar webpage to register for this and other future webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would really appreciate it if you can please take a moment to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you for your time.